Hello, everyone. I would like to welcome you. I'm Patrick Schultz, representing the Instruct Centre FR1 from Strasbourg, located in the Center of Integrative Biology in Strasbourg, France. We have today the privilege to host the 10th issue of the Instruct webinar series, Structure Meets Function. This series of seminars gives a great overview on the achievements and the challenges of integrative structural biology. It emphasizes the need to have access to high-end technology, to combine various methodological approaches, and to engage innovative developments, and this to address complex biological questions. We have a rather tight program today. I will first ask Bruno Klaholz, who is the head of the infrastructure here in Strasbourg, the Instruct-ERIC Centre FR1, to give a brief overview on the specificities of our center, the ongoing developments, and to highlight some scientific achievements. And we will then continue with three presentations from PIs of the Integrative Structural Biology Department here at IGBMC in Strasbourg. You will be able to ask questions at any time, either by using the Q&A box, or if you want to ask the question directly, to raise your hand, use this raise hand option, and you will be given the mic after the talks. So I would also like very briefly, so that I don't forget it, introduce the next webinar, which will be in June, early June, the 8th of June, and which will happen in the Instruct Centre France 2 in Grenoble, with three talks also from Franck Fieschi, Martin Weig, and Hélène Mallet. The session is open for registration already. So with this, let's start, and I will give the mic to Bruno Klaholz to present the center.
Okay, good. So many thanks, Patrick. I'd like to give an overview indeed on the CBI, the Center for Integrative Biology, and the infrastructures that it hosts. So we have Instruct-ERIC represented nicely today, the national infrastructure Frisbee, and INEX Discovery as a European infrastructure. The CBI itself has a number of driving scientific projects around transcription, translation, RNA, which involves many nucleoprotein complexes and biomedical targets. And regarding the infrastructures that it hosts, Frisbee is a national infrastructure that comprises 5 centers, among which 2 are Instruct centers, so Strasbourg and Grenoble. And Instruct as an infrastructure provides user access, for example, for research visits, for training, for internships, and also for R&D funding, which you can apply for on a regular basis. It comprises now 15 members across Europe. And the recent call was just launched actually yesterday for Instruct for COVID-19 projects. And there's also one running in INEX Discovery and in Frisbee for those interested to apply. What we offer at the CBI in Strasbourg is protein production facilities, mammalian cell expression and vaccinia systems, for example, as rather specific systems, which are quite asked for, quite often as well. Biophysical characterization of samples, crystallization facilities, X-ray crystallography, NMR, cryo-EM and electron tomography, super resolution fluorescence microscopy, Histone basically, and dedicated computing resources for processing. And the platform has dedicated engineers and is strongly supported by associated research teams. The platform comprises modules for each of these activities that I just mentioned, where you can get various projects done and get support and also training for the users when they come outside. And in terms of training, there's also a national school that was set up and coordinated by Valérie here at the center. That's happening every year. Next one is actually in June. That is training across the various techniques in structural biology. In terms of R&D at the CBI, we're strongly investing in cryo-electron microscopy as a flagship for Instruct. And here are the latest results showing this is work on the Titan cryos with apophreatine test data going to basically 2 angstrom right now. And the point here is that we do that with on-the-fly processing, which involves movie alignment, CTF correction, and particle selection, which goes within a few hours to get a first classification and a reconstruction a bit later. So that during data collection, you can decide whether you want to continue or if it's better to change sample, or if you have already enough data and you want to change sample to collect other data. There's various possibilities. So that's very important actually to have scientific computing and the developments around that. The glass-fused microscope that was just installed very recently has, in addition, a specific setup to do microelectron diffraction with a ZD camera, which is optimized for diffraction, and the data collection with EPUD. Okay, there's also another aspect that I would like to highlight here for those interested is super-resolution microscopy, which is really bridging scales within structural biology. And what we have done over the past years is actually develop softwares also to improve alignments and resolution for super resolution imaging with various tools, including with user interfaces. And the latest developments are actually allowing to do that directly in 3D. So you can do 3D segmentation, and we're also working now on multiple labeling approaches. So that's really in the context of integration scales of various techniques that are all on site here. And that is really the move of the entire field, as you know. Basically here you see the statistics of PDB depositions over the last couple of years. Since resolution revolution in electron microscopy, thanks to detectors and image processing methods, the number of high resolution structures is increasing. And what you can also see, and that's in this paper if you like to check, essentially all large molecular complexes are now studied by electron microscopy. It's also, and with a comparable resolution, whether it's done by X-ray crystallography phasing techniques or directly by 3D reconstruction from cryo-EM images. So in that context, we have set up a roadmap in France to strengthen cryo-EM, and in Strasbourg, this is really building up a national center for biomedical cryo-EM. That facility will comprise several high-end and screening electron microscopes, implementation of new cryo-cooling devices like the Chameleon is to come in the next few months, super resolution instrumentation and corrective imaging, also fibbing, so cutting sections, cellular sections for cryo-electron tomography, sample preparation, crystallization, micro-ED, as I just mentioned, dedicated computing on the fly processing. So this is to come on at that center to be hosted at the CBI, at the GBMC campus, and part of the 3 infrastructures, Frisbee, Instruct-ERIC, and Inex Discovery, with 24/7 access like at synchrotrons, project-based. So you see the history of the site in terms of high-end instrumentation and electron microscopy, for example. Here, Polara installation, the first one in France, it's time. Titan cryos also the first one, with latest updates and upgrades in terms of faceplates and cameras, for example, the K2 and then the K3 camera that is now on, and the Falcon 3 at the same time. The glass-fused installation that has a K2, and there's the whole setup from a device to do lamella preparation and SEM imaging. So there are a number of results from the site, and I will just illustrate a few from my own group. We've been working on water faceplate enhancement methods to really get image contrast that is improved and allows to actually improve the data processing up to the 3D reconstruction of complexes. We also worked on the image processing approaches, including focus classifications early on here in these data, and continued then on recent data for focus classifications and refinements. And that's really essential, because with that we have been able to determine the structure of the human ribosome a few years ago, then form complexes with drugs and analyze them by cryo-EM. So basically it becomes feasible to analyze drug complexes by cryo-EM and push resolution to even see chemical modifications. And the latest data are here on an mRNA complex mechanism that will be published soon, I hope, on the Cossack mRNA recognition, which is an important eukaryotic mechanism. Lately, that's a work in press now in Science. It's a mechanism of a drug target for Rett syndrome, where we could determine the crystal structure of a complex and showing the specificity of modifications of the DNA being recognized by one given residue that is actually mutated in the Rett syndrome. Yeah, so with that, I'd like to hand over to the next speakers and illustrate more of the results of the CBI over the last couple of years. So any questions, welcome, please.
Thank you, Bruno, for this brief presentation of the center and of the highlights of your group. Let's move on with our program. And the next speaker will be Valérie Lamour. Valérie has a background in chemistry, biochemistry and structural biology. She graduated in Dino Morat's lab and defended her work on gyrase-B and X-ray crystallography. After 3 years postdoc at Rockefeller University in the lab of Seth Darst, she accepted an assistant professor position in Strasbourg Medical School and started her own team. She will now show her latest results on the structural organization and the allosteric regulation of human topoisomerase 2 alpha. Please, Valérie, the floor is yours.
Thank you, Patrick, and thank you for giving me the opportunity to present our latest results on the structure function analysis of the human topoisomerase. So maybe just a short introduction on DNA topoisomerases for those who may not know about these enzymes. They're evolutionary conserved enzymes from bacteria to human, and they're essential proteins that release DNA topological stress that's induced during cellular processes, and in particular during replication and transcription, where accumulation of supercoils may prevent the progression of the machineries on the DNA. Also, during chromosome segregation, there's several topological structures such as catenates require the activity of DNA topoisomerase, and in bacteria, DNA compaction of the bacterial genome requires also the activities of the topoisomerase. There are two classes that have been discovered in the 70s, the type 1 and type 2 DNA topoisomerase, and they all share the same mechanism of cutting the DNA and religating in order to release torsional tensions in the DNA molecule. In human, there are two isoforms, the TOP2 alpha and TOP2 beta. TOP2 alpha is overexpressed during mitosis and is important for replication, essential for cell division, and because it's overexpressed in cell proliferation, it's a biomarker for cancer. The TOP2 beta was discovered much later and has been shown to be involved in activated transcription complexes and important for transcription of neuronal genes. Defects in these proteins or in the repair of the cleavage DNA breaks of the TOPO2 can lead to cancer, and more recently it's been identified to be involved in processes leading to neurodegenerative disease. The TOP2 alpha and beta are highly conserved on the catalytic domain. They're composed of an ATPase domain that binds two ATP molecules, a DNA binding cleavage domain, and together they form like a large modular homodimer of about 350 kilodalton. Like all DNA topoisomerases, they have a conserved tyrosine that is catalyzing the DNA break at the 5' end of the DNA molecule. So since these enzymes are dimers, they induce a double strand break in DNA. So the alpha and the beta isoform mostly differ in their C-terminal domain that is highly predicted as highly disordered and has no sequence homology with each other. Another level of regulation for this enzyme is the presence of post-translational modification that accounts for some of their specificities. So the accumulation of biochemical data over the past 40 years and 3D structure of individual domains have led to propose a 3-gate mechanism in which the enzyme binds at the dimeric interface a first DNA fragment called the G segment, and upon binding of 2 ATP molecules, a second segment of DNA is trapped in the protein, forming a DNA crossover as observed in the DNA supercoils. And after cleavage, double strand break of the first DNA, ATP hydrolysis triggers the DNA transport of the second strand. The hydrolysis of a second ATP leads to resetting of the enzyme that is primed for another cycle. Several therapeutic strategies to inhibit these enzymes were developed, and the most successful one is the one that targets the DNA cleavage complex with molecules that intercalate in DNA, such as etoposide or doxorubicin that are used in cancer treatments. And the goal of this allosteric cycle is very tightly regulated, and topoisomerase ensures that there's no DNA breaks. And of course, upon the addition of intercalants, the goal is to lead the proliferating cells to cell death. So as you've seen, there were at this stage only individual domains that were solved. And the question to understand the allosteric mechanism of this enzyme and how the domains are organized and coordinated is to be able to get the full architecture of the nucleoprotein complex. And even if those enzymes share a great deal of homology on their catalytic domain, the devil is in the details, and a lot of the molecular determinants lies into the connection between the domains. And also there's so far little information on the implication of the Cterminal domains of these enzymes in the allosteric regulation. So to stabilize these modular and highly flexible proteins, we're using small molecules to lock in conformations. So we started a couple of years ago on the bacterial system DNH arrays, and the first result we got at the time using the Polara microscope with a CCD camera, we're able to get the first reconstruction of the Thermus germophilus DNH arrays, and we could show that subunits of the topo2 are intertwined, which has consequences on the mechanism for trapping the DNA crossover. And resolution revolution later, we were able to get a reconstruction of the E. coli DNH arrays, which is a genetic model for all of the studies in the field. And with this structure, we could analyze all of the connection between the domain for the bacterial enzyme. But of course, the human topoisomerase is another level of complexity. It's much more difficult to manipulate. So for this, we had to set up specific tools to get large amount of protein amenable to structural studies. So we initially started with producing the protein in yeast, but we quickly were concerned about the state of the post-translational modification. So we moved to a system that was set up by Robert Drian and Karine Prado in the lab, using mammalian cells overexpression system that takes advantage of modified vaccinia virus that's been evolved to solely infect baby hamster kidney cells. So we actually co-transferred a vector containing our gene of interest together with the infection with the parental virus, and to generate a recombinant virus. So the innovation in this system was that to speed up the selection process of the recombinant virus, Robert and Karine introduced in the parental virus M-cherry and a fusion with gyrase and terminus with kinase that ensures that we can select quickly after recombination for the recombinant virus. So we monitored the loss of M-Sherry, and adding coumaricin and mycophenolic acid ensures that we get quickly the recombinant virus. So with these three different selections, the process that used to take several months was speed up to 2-3 weeks. And after we get a recombinant virus, we can infect large volumes, 6 to 8 liters of cells. And in 4 days, we can then extract the topo2 alpha. And depending on the preps, we get between 5 and 10 milligrams of human topo2 alpha. So with this protein, Arno van der Breck, a former PhD student in the lab, optimized the complex formation, and we formed a nucleoprotein complex with doubly nicked oligonucleotide of 30 base pair, and that we added the etoposide, which is this cancer drug that intercalates into the DNA breaks. So using the Titan cryos, he could collect several data sets and did a reconstruction, and you can see here the composite cryo-EM marks that illustrate well how ordered the DNA binding cleavage domain is compared to the ATPase domain. And this flexibility that we observed already in the bacterial enzyme, we can see already at the level of the 2D classes. However, using strategies of 3D classification and refinement, it could get better resolution on this DNA binding cleavage, and we can observe the etoposide molecules that's intercalating between base pairs in this region with local resolution at 3.6 angstrom. Still using these strategies of 3D classification, Arno could correlate two conformations in the DNA binding cleavage that with the positions of the ATPase domain. And with this, he was able to reconstruct two conformational states of the human topo2, a closed and a pre-open complex. Pre-open meaning that the DNA is opening despite of the fact that we added some drugs to lock in the DNA. There's still a conformational fluctuation around these two positions that prefigures how the DNA gate is opening during the cycle. So what was interesting is that we could, for the first time, observe the transducer releases that adopt a different orientation than in the bacterial enzyme. And Christophe in the lab was able to test the different mutants of this transducer and show that they have an importance. The specific eukaryotic motif are important for the allosteric communication. One of the striking features of the structures is that despite of the fact that we used the full-length protein, we were not able to locate completely the C-terminal domain. However, since we use a full-length protein, we still could see like the linker linking to this C-terminal domain. This is quite important because most of the constructs that were used to solve structure of the DNA binding domain were limited to this position here at the residue 1191. And so we explored this linker to the CTD. And by doing some deletion mutant, to our surprise, this construct with this linker showed an increased activity compared with the wild type containing the whole CTD. So when we look at the structure, we can see this linker goes below the DNA and helps stabilize the curvature of the DNA that is necessary for DNA cleavage. So we think that the role of this linker is to stabilize the DNA curvature for cleavage and then for DNA relaxation. And this also means that the bulk of the CTD is actually down-regulating this allosteric effect. And this is quite interesting because in this region, we can observe post-translational modification. In particular, there's a position at residue 1213 that is known to be phosphorylated and trigger the decatenation checkpoint. And a lot of modification are located on this C-terminal domain. And so we think that post-translational modification probably adds another level of allosteric regulation. And maybe just as a perspective to finish, so with these results, we analyze all of the post-translational modification, phosphorylation and acetylation on the protein that we produce in yeast and in mammalian cells. And we provide now for the community of topoisomerase research all of the PTM sites in non-cancer cells. And we could, when we map them on the structure, we can see that acetylation are clustered on the catalytic sites. And the phosphorylation are mostly clustered on the C-terminal domain. So we've analyzed a couple of these positions already, and we can see some interesting features and how this post-translational modification actually impact the catalytic activity. What's interesting also is that it's been reported in the literature that post-translational modification are correlated to some drug resistance. And this is surely something we would like to explore now using the mammalian cell system. And to finish, I think we can say that now this reconstruction of the topo2 alpha for the first time provides a platform for analyzing all this regulation, and in particular, the next level of regulation, which is all the post-translational modification. And with this, I would like to thank all the people in the lab, in particular Arno van der Broek, a very talented PhD student who is now doing a postdoc at Rockefeller with an EMBO fellowship, Christophe, who did a lot of the enzymatic assays, and Robert with Karine, who developed the mammalian system with the vaccinia. I would like to thank also all the people working on the cryo-EM facilities, the CBI supported by Instruct and iNext, in particular Julio Ortiz, and now Alexandre Durand and Niels, and members of the Schultz and Klaholz team who are constantly supporting all of the cryo-EM infrastructure, Catherine Birk at the protein production, and the IGBMC common facilities, Betty at the cell culture, and Luc Necroni for the mass spectrometry. And I thank you for your attention.
Yeah, thank you for this talk, beautiful talk, Valérie. We should perhaps start with some questions from the audience. So maybe I start with one. I mean, all this is about movements and conformational changes. So how do you think we could address this in more details in the future, especially if we have continuous movements? Is it worth combining this data with thread data, for example, to get deeper insight into the conformational changes?
Yeah, it's a good point. There are people in the field working with threads. The difficulty with the human protein is it's not as easy as the bacterial protein to manipulate in different techniques. I think also what would be interesting is to go to larger complexes, because the conformation we observed in vitro and isolated complexes, we're trying our best to stabilize some of those conformations, but it would be useful, I guess, to go to the next level, to the chromatin level, to be able to make sense of this conformation in the chromatin complex. So in solution, studies are complementary to what we do in cryo-EM, but I think what would be more relevant is actually trying to observe what are the relevant conformation in the context of macromolecular complex, larger macromolecular complexes.
Okay. So there is a question from Adriana Erika Mille. Maybe I just read the questions with the two conformations in hand. do you think that drugs that will prevent the movement could be more effective and specific of DNA as compared to DNA intercalating drugs?
Yeah, it's a good point. I think now that we have these two confirmation, we could definitely explore several pockets that are revealed by this different conformation and try to potentiate those intercalating drugs with other molecules. So this is clearly one of the interesting follow-up of these structures that are now out there and that can provide information for drug design programs. Yes.
Mm-hmm. There's another question from Caroline Austin, who asked whether the human alpha, very specific question, contains the 26 first amino acids.
Yes. So those are the construct we've expressed in the mammalian cells. We reintroduce all of the 26 amino acids that very often in the field were removed in the initial yeast construct. So yes, it contains
a very N-terminal part. This is where the
yeah, this is at N-terminus of the ATPase domain, and so this is a non-conserved tail that might play a role in coordinating some of the steps, but there is little information about this right now, and we don't see it in the cryo-EM map. It seems like it's disordered.
Stephen Cusack has a question also about the fact that the protein is a heterodimer, and does the cleavage occur twice or only once in the complex? And if it is only once, how was the active site, the cleavage site, identified?
So it's a double strand break because the way the protein is working is by a nucleophilic attack of tyrosine on the phosphate backbone of the DNA. So since it's a dimer, there are two tyrosines, and so it's a double strand break. And you can actually monitor the double strand break using a very simple assay on an agarose gel, where you can visualize the transition from circular DNA or supercoiled DNA to a linear cleaved DNA.
So Eva Hyde was asking whether you can possibly see additional intermediate states by using other drugs.
It's a good point, yes. We believe so. The thing is, these assemblies have been difficult to tackle using structural biology because of the extreme flexibility of the domains. And when you see the mechanism, it makes sense that all of this is moving to be able to transport the DNA all throughout the dimeric interface. There are several compounds that can be used to block different regions of the protein. So by using a non-naturalizable homologue of ATP, ADP, and P, we locked in the ATPase domain. And with the toposide, we're locking in the cleavage complex. So we locked at two different positions. And we could imagine using other drugs to explore other conformation. This is certainly something that would be interesting to do.
In the same line of questions, Jean-Baptiste Chamollier asked a question about do you now understand how a toposide inhibits the topo? And do you have only one, or do you have two toposide molecules in the group?
We actually have two. I only showed the density of one. The two sites are, the two molecules are visible in the structure. One of the sides is a bit more defined than the other one, but there are two molecules are intercalating in the two cleavage sites.
Mm-hmm. Again, another drug. Caroline Austin also asked whether you plan to analyze ECRF 187.
Yes. That's a good point. That would be really interesting to do. We haven't done that yet, but that would be something interesting, yes. And
Nikos has a more general question, given that enzyme proteins in general adopt an ensemble of conformations. Do you time freezing a single average conformation if other conformation exists, how different are they and how do you address this issue?
How different are they? So again, it depends how we form the complex. I think this is highly dependent on the components we put in the complex, the DNA, the small molecules. So to be able to get the best reconstruction possible, because our goal, our first goal was to get the full architecture and to be able to visualize the connection between the domains. And so in that sense, we try to lock in as much as possible the enzyme. Still, we see some conformational fluctuation. There are many more conformations that we could observe in the 2D classes, but we went for the most populated populations to get the better resolution. We would need to collect much more data, and as it was mentioned before, probably try to form different complexes with different DNA templates, different lengths of DNA, and different small molecules, so we can achieve and reach to the full conformational landscape of this molecule.
Okay, thank you again, Valérie, for this great talk. You are afraid of being a little bit rusted after the pandemics, but no, be sure everything is fine.
Thank you.
The next speaker is Gabor Papay. Gabor first studied the transcription regulation in Drosophila embryos in Imre Boros's lab in Szeged, Hungary, before switching to structural biology and applying single particle cryo-EM approaches to the study of the structural organization and the function of transcriptional co-activators. These co-activators integrate cellular signaling, chromatin structure, and epigenetic changes to favor transcription initiation. He will present today's work on the Saga co-activator, and in particular how it holds and deposits the TBP, the Tata box binding protein, in its structure. So please, Gabor, the floor is yours.
So now, as Patrick said, I will talk about the structure of the yeast Saga transcriptional co-activator. So first of all, I would like to do a brief introduction about what is a transcriptional co-activator. So in eukaryotes, and now this is adapted, this figure, to yeast, the transcription is initiated by by an activator which will bind to its target site. And then it has to free up the transcription initiation site. Most of the cases, there is a TA-rich sequence, which is called the Tata box or Tata-like sequence, where Tata box binding protein and TBP will bind, and then this protein will recruit the transcription pre-initiation complex, which contains the RNA polymerase II, and then it will transcribe the gene. So all this is kind of concerted by the transcriptional co-activators. So here there is the Saga co-activator. This can interact with the activators. So after the activator is recruited, it can interact with Saga. And Saga helps to recruit chromatin remodelers, which can alter the chromatin landscape, remove nucleosomes or slide nucleosomes, and also can read and write histones of the nucleosomes. And it also can interact with the TBP to stimulate the pre-initiation complex formation. The Saga is a large protein or protein complex. It's almost 2 megadalton, and it has 19 subunits. It is conserved throughout the evolution, and it has 4 main functional modules. One is the het activity module, which can acetylate the histone H3. And there is a deubiquitination module, which can remove a ubiquitin from the histone H2B. There is the TBP binding module, which can interact with the TBP. And there is a large protein, it's more than 400 kilodalton, which is called TRA1. It is the part of it's a PAKK kinase family. And this was said to be responsible for the activator binding and the interaction with the activators. It also contains several other subunits, which has a structural role, but I will show you later. So it's not only Saga which interacts with TBP. There is another complex which is called TF2D. And together, these two complexes require on all gene promoters for an mRNA expression. The two complexes share several subunits, and these subunits contain a domain homologous to histones, and they can form specific heterodimers. And TF2D contains a stoichiometric amount of TBP, but Saga is not. So when we purify Saga, it contains just a little TBP, but we can form a stable complex with if we provide an external TBP. So our team is working on Saga since 20 years now, and there are certain obstacles to handle. So the main obstacle is that Saga, as you see here, is not very abundant. So if you look at one yeast cell, you will have around 200,000 ribosomes, around 30,000 of polymerases, RNA polymerase 2, and the co-activators are much less abundant. So the mediator is around 6,000, but Saga is only 1/100th of the ribosomes. So we, throughout the years, we try to improve our biochemistry and all the workflow to have higher and higher resolution of the complex. So the biochemistry was improved by Adam Benchem. He decided to use another yeast, which is the Pichia pastoris, which can grow to higher density than the Saccharomyces cerevisiae, so we will get more protein. He isolates the nuclei to reduce the exposition to the cytoplasmic proteases. And then we played a lot with the buffer composition, detergent concentration, and with various cross-linking methods. We ended up with cross-link with glutaraldehyde, and we optimized a lot how we vitrify the sample, so how we prepare the grids and then vitrify the sample on the grid. And to obtain the latest structure what we had, the instrumentation was also changed a lot. We started with 120 kV Philips microscope, and then at the end we used the Titan cryos with the direct detectors instead of the films. And these results are much better quality of the data. And to deal with the data, we also took advantage of the new developments in the softwares. So this is the structure what we obtained. We formed the complex with TBP, and here you see the structure with two different orientations. This is the modules are here. This is the TRA1 part. It's just the huge protein which occupies the half of the structure, the densities. And here you see that the enzymatic modules, the histone acetyltransferase module and the deubiquitination module are really not well resolved, so they are very, very flexible. And the TBP site was also quite flexible. So this is the model what we created, and then I would just dive into the model and look at or show you some details of it. First of all, how the enzymatic modules are attached to Saga. So we don't see really the enzymatic modules because they are very flexible, but we see the attachment sites. And this is the protein SGF73 is part of the deubiquitination module, and it is integrated into the complex, where you see it's in the upper part, which I would call a main module. It's really intercalated inside the different proteins. And the other one is the histone acetyltransferase module. What we know that it is on the top of the structure, and we, based on cross-linking data, we attributed the last 3 or 4 helices to one of its subunit, which is called ADAS3. And so we see how it interacts with the rest of the complex. So this is the main module. So here you have the TRA1, and this is the upper part. And here we have the control module, which would say it's stuff 5, and several subunits, which is called TUFs, which are shared with TF2D. And then here you have the histone fold module, which is also part of the TUFs and shared with TF2D, and then the TBP binding module. So this main lobe is organized by one domain of one of the subunits called TUF5. It's a WD-40 7-propeller blade, and this interacts with the rest of the complex. So it connects the TUF6 and TUF5 together. This is important because the position of these two modules in the other complex TF2D is very different. So this probably ensures that from this building block there will be a Saga build. Then it also interacts with the enzymatic modules, and it keeps the histone fold modules together, which at the end will interact with TBP. So this WD-40 module, just zoom on it, it interacts with 11 different protein domains, and this is part of seven different proteins. So it's really an interaction hub of the complex. So I come back to the histone fold part. So this is which interacts with the WD-40 module and then goes towards the TBP interaction site. So this histone fold part is really reminiscent of the nucleosome. So here you have a nucleosome with the histones. It's a histone octamer, and if you compare the histone fold octamer in Saga, this has the same or very similar structure. Here we have the subunits with the histone fold domains in Saga, and these 8 histone fold domains will form a histone fold octamer. But there are certain differences if we compare it with the nucleosome. So the SPT3, which will interact with TBP, has a much weaker interaction with the rest of the octamer than in the nucleosome. And for this, you will see that the SPT3 will kind of flip out of the octamer, and this will help to accommodate this. Also, you will see that it starts to deviate from the histones, and this will help to accommodate TBP. Another difference is that the C-terminal tail of SPT3 will penetrate into the middle of the histone fold octamer, and it interacts with all of the histone fold containing proteins there, and then thereby stabilizes the structure. Then, and at the other part of the SPT3, there is the interaction with TBP. So SPT3, which interacts with TBP, and the other side, there is another protein which is very dynamic and very flexible. We couldn't really resolve it in the structure, which is called SPT8. So this is connected, this SPT8, with the 2 long helices of another subunit SPT7. And this we know because if we delete this part of SPT7, then SPT8 is not purified anymore with Saga. There is a block here of SPT3. There's a helix which blocks the DNA binding site of TBP. And here you see a blow up of the interaction site, which shows that our TBP has a pseudo 2-fold symmetry. It has it's like a saddle with 2 stirrups. So one is the N-terminal stirrup and one is the C-terminal stirrup. And the hydrophobicity of the stirrups are different, and the pocket will choose. So it is specific to one of the stirrups. So TBP is really specifically interacting with SPT3. And this interaction is similar to TF2 with another protein interaction with TBP, which is TF2B. So if you look at the structure itself, and we will try to fit a DNA, a promoter DNA, we know the structure of the TBP interacting with the DNA, then what we immediately see that this interaction is blocked by a steric hindrance of the TRA1 protein. So if you look at then Saga and TBP cannot interact with the DNA. And if we add TF2A, then it can immediately interact with the TBP, so with the DNA. So here you have the free DNA, and this is an electromobility shift assay. And here you have when the DNA is shifted, when it interacts with the protein. So there should appear here somewhere, but I don't see a frame showing that Saga and TBP does not interact with the DNA. But if you add TF2A, then you will have the interaction immediately. This is with the DNA which contains the Tata box. If we try to do with the DNA without the Tata box, then we don't have this interaction, or it's very, very weak. If you have a kind of a mutation between the Tata box, it's like a Tata-like sequence, then we have a stronger interaction with the DNA. This shows that this complex here has a specificity for a DNA which contains a Tata box or contains a TA-rich sequence. This was also shown by other labs with other meanings. So this actually at the end, what we can say that Saga holds TBP, and as TBP was shown that if it's free, it dimerizes, so it completely blocks the DNA binding surface. So like this, TBP is monomeric, and the DNA binding surface is more exposed. And if another complex TF2A is coming, which is part of the pre-initiation complex, then it can interact with TBP and then will be released from Saga, and then the transcription can start. So there is a little comparison with TF2D and Saga. So with Saga, as I showed you, there is a histone fold octamer, but in TF2D, there is a histone fold hexamer, and somewhere there, a histone fold octamer. This was, we had a low resolution part of this lobe, so we couldn't really track the histone fold octamer of TF2D, but as these are homologs, so we thought that they could be very similar. And just recently, there was a structure published of TF2D where they could resolve this part, and then if you compare this to Saga in blue and TF2D in yellow, then you will see roughly that they are very, very similar. So the mechanism of TBP release, at least in yeast, should be very similar in TF2D and in Saga. And at the end, I would like to thank people from the Patrick Schultz lab who helped us to make this structure. So the biochemistry was done by Adam Bensham, and the first structures was done by Grigory Sharov, and also Olga Kolesnikova was helping Adam for the biochemistry, and Alexandre Fréchard was also helping in the structure building. And thank you for your attention.
Thank you, Gabor, for this presentation. The session is open for questions and discussions. What are your perspectives, Gabor, to try to stabilize the enzymatic modules, which are still very mobile and for which we don't have any decent structure right now?
Yeah, so we are trying to do some interactions with the enzymatic modules. First of all, we do kind of using a reagent which interacts with both of the enzymatic modules, and then maybe they will be logged. But frankly, the histone acetate transferase module is very, very flexible, and it will be really hard to stabilize it.
So Caroline is asking whether other complexes bind TBP.
Oh yes, a lot of complexes bind to TBP. There are not only the RNA polymerase II machinery, but there is RNA polymerase III and 1 machinery. And TBP is uniform, so TBP is the most important protein to start transcription. So for starting transcription in eukaryotes, you need TBP. So then all the machineries which will bind to it. And there are also other complexes which will remove TBP from the DNA. So there are also complexes which, like TAF1B or the others, which will then interact and remove the TBP from the DNA.
Jean-Baptiste Charbonnier had a question about yeast genetics and whether all the subunits are essential in yeast, and perhaps you can also say something about the human complexes.
So no, the yeast in yeast, Saga is not really essential. GCM5 is essential. So this is the histone acid transferase enzyme, but the rest of the subunits are kind of the yeast can survive. Saga was mainly described to help in stress conditions. So we also use a kind of a bit of a stress to enrich Saga in our purifications. And but the rest of the subunits, except for the TAFs, so the subunits which are shared with TF2D, they are essential. TF2D is an essential complex. So all these TAFs, which are the TBP associated factors, they are essential. In human, it's a bit. So the yeast has only one. In the human, there are many more complexes, which not many more, but more complexes which contain GCM5, and they share functions. But last Lotora was showing that if you delete the yeast and also the human Saga subunits, then the newly synthesized RNAs are dropping. So this is why he concluded that Saga is needed for all of the transcription.
Any additional burning question? Otherwise, we'll switch to our last speaker of this session, Albert Weixlbaumer. He studied microbiology and the genetics in Vienna and got interested in X-ray crystallography during his PhD in Venki Ramakrishna's team at the MRC, Cambridge University, where he determined the structural functional states of the ribosome. He then moved to Rockefeller University to join the Seth Das group and studied transcriptional pausing events, looking at RNA polymerases by X-ray crystallography. And with a rather high degree of consistency, Albert will now present his latest work on how transcription and translation are coupled and how RNA polymerases and ribosomes deal with each other. Please.
Thank you very much. I hope you can all hear me. So yes, my name is Albert Weixlbaumer, and I would like to tell you what we have learned from our reconstructions of a bacterial, which is a complex of a transcribing RNA polymerase shown in red and a translating ribosome shown in yellow and blue here, which we published last year. So let me start with a brief introduction. So this is a Miller spread that I'm sure most of you have seen in one form or another, and it depicts the entire process of prokaryotic gene expression. So you have the RNA polymerase, which transcribes DNA to RNA, highlighted with an arrow here. And as transcription progresses, you can see that the spherical particles join RNA polymerase, which of course have been interpreted as being ribosomes that translate the newly synthesized mRNA as it emerges from the polymerase. And so this picture actually was the first, or one of the first, visual confirmations of a proposal that had been made by Marshall Nirenberg in the mid-60s, namely that transcription and translation are coupled processes in prokaryotes. And over the decades since these early discoveries, people have made numerous contributions that suggested that transcription and translation are kinetically coupled, meaning their rates match each other in vivo. Although it's important to realize that in some species, for example, Bacillus subtilis, this does not seem to be the case. Going further, people have also suggested that transcription and translation are not just kinetically coupled, but also physically coupled. And this was shown, for example, in 2010 by Paul Rösch's lab, when they published an NMR structure of a transcription factor called NUS-G, shown in green here, bound to a second transcription factor called NUS-E. Now, the ribosome community knows NUS-E better under its alter name, which is US10, and it's part of the small ribosomal subunit. And what Paul Rösch's lab suggested was that NUS-G, which uses its N-terminal domain to bind RNA polymerase, and which can use its C-terminal domain to recruit transcription factors, including US10, may actually form a physical bridge, a tether, if you wish, between the transcribing RNA polymerase and the translating ribosome. And this idea got further support a few years ago when Max Gottesman's lab showed that mutating residues at the very interface between NUS-G and NUS-E abolished NUS-G co-purification with ribosomes. However, this view was challenged when in 2017, 2 low to medium resolution cryo-EM reconstructions were published. The first one from Andrei Korostelev's lab, which was a complex of the small ribosomal subunit shown in yellow here with RNA polymerase, where the polymerase bound at the mRNA exit site of the 30S subunit. And then in the same year, Patrick Kramer, in collaboration with Bob Lendig, published a reconstruction of polymerase bound to a 70S ribosome. Now, this complex had been formed by letting the ribosome collide with a stalled RNA polymerase. And in this reconstruction, the polymerase bound to the mRNA entry site on the 30S subunit, as you would expect. It's also important to note that the Kramer reconstruction was actually confirmed in situ directly last year from tomography work that was done in Julia Mohamed's lab in collaboration with Yuri Rapsilba. So these 2 reconstructions showed inconsistent ribosome binding sites, and they also challenged the NUS-G coupling model, because if you look at the binding sites for the NUS-G N-terminal domains on the polymerase, and you measure the distance to the binding site for the NUS-G C-terminal domain on the ribosome, you will see that they are too far apart. And therefore, in these configurations, NUS-G would not be able to form a physical link. So around that time, we also decided to address this problem. And in particular, we wanted to understand, why do we see additional or alternative RNA polymerase binding site? We wanted to know what drives expressome formation. Is it direct interactions between the polymerase and the ribosome, or is it something else? And then we wanted to gain some insights on potential functional roles of coupling between transcription and translation. And finally, we wanted to understand if she is able to physically couple the 2 machineries. And so we used a different approach in the sense that we assemble expressomes from purified components using mRNA substrates that direct binding of ribosomes and RNA polymerase. And by varying the spacing between the 2 binding sites, we can mimic a ribosome that approaches a transcribing RNA polymerase. And I would also like to emphasize that a similar approach has been taken by Richard Ebright's lab, and they came to very similar conclusions, and in fact published back-to-back with us. So using this approach, we first obtained preliminary reconstructions that showed 2 fuzzy clouds that in fact matched the 2 RNA polymerase binding sites that had been identified by the Korostelev and Kramer lab. But I can't go into too much details, but what we found is if we saturate our ribosomes with a protein, ribosomal protein BS1, we can abolish binding of RNA polymerase to this position. And it's important to note that BS1 tends to dissociate during ribosome purification. So by saturating the purified ribosomes with BS1, we could get rid of this polymerase binding site. The question also arises, what drives expressome formation? And so we found that in our hands, there are no direct contacts that drive expressome formation. Instead, if we use mRNAs that are long enough to support binding of both polymerase and ribosome, we can see co-purification of the polymerase, showing as this doublet here with ribosomal proteins. But if we use a short mRNA or no mRNA at all, then this stable complex formation is in fact abolished, and we do no longer see co-purification. So using this approach, we moved on and evaluated the compositional heterogeneity of our complexes. And Michael Webster, a postdoc in my lab, found that only about between 10 to 20% of ribosomes contain RNA polymerase bound to it. And so this, posed a significant problem in terms of the amount of data that we needed to collect. And so what Michael did was, so this is true both for negative stain and cryo conditions. And so what Michael did, he used his data processing skills and first selected particles based on the ribosome, and you can see weak density corresponding to RNA polymerase. He then re-extracted those particles, but now centered on the RNA polymerase density, subtracted the ribosome signal, and then performed another round of 2D classification to enrich particles that really contain RNA polymerase, and then reversed the signal subtraction to now obtain a better reconstruction with improved density for RNA polymerase. But as you can see, it's still essentially a featureless blob. And so you can probably more appreciate this in 2D classes that are either aligned to the ribosome, where the polymerase is a fuzzy cloud, or aligned to the polymerase where the ribosome is a fuzzy cloud. And this highlights the flexibility that is inherent to this complex that we think represents an uncoupled expressome, where the only consistent connection between the 2 machineries is the shared mRNA. Nevertheless, using focused refinement approaches, Michael was able to obtain a high-resolution composite EM map that allowed us to build an atomic model of the entire assembly. And so this is the first time that we can see at high resolution the entire process of gene expression within one molecular assembly line, starting with RNA transcription in the active site of the polymerase, going to the decoding center of the 30th ribosomal subunit, and ending at the peptidyl transferase center in the 50th subunit, where the incoming amino acid is highlighted in red here. The maps really were of superb quality. We could see modifications on ribosomal RNA residues, and our ribosomes typically refined to about 3 angstrom nominal resolution with higher resolution in the core, and our best polymerase reconstruction refined to about 3.8 angstrom overall resolution, again with high resolution in the core. So using this focused refinement approach, Michael also developed a new approach to essentially measure the conformational heterogeneity that is inherent to our complex, because conventional 3D classification or multi-body refinements did not allow us to obtain high resolution reconstructions. And so what Michael did is using angular assignment for ribosome and using the angular assignment for RNA polymerase, he was able to calculate for each individual particle what is the relative orientation between the two machineries. And plotting this, on the x-axis, you essentially see rotation around a rotation of polymerase around an axis that is essentially parallel to the mRNA, and around the y-axis, you see rotation around the perpendicular axis. So using this approach, Michael could see that the polymerase is not adopting random orientations, but instead it clusters in particular positions. And so he could then select particles corresponding to these clusters and obtain lower resolution reconstructions, because the number of particles is now much smaller, but nevertheless, it allowed him to rigid body dock RNA polymerase and basically confirm that this approach is working. But most importantly, in one of these clusters, we observed additional density that matched where we expected the NUS-G N-terminal domain to bind RNA polymerase. And so it also turns out that in this orientation, the polymerase would be able to simultaneously bind to the NUS-G NTD, while the C-terminal domain of NUS-G would bind to the ribosome. And so using this knowledge, we then moved on to form a sample that we call the NUS-G coupled expressome, where we now added NUS-G at saturating amounts after purifying the expressome, and we obtained now a reconstruction with much better defined RNA polymerase density, where conventional 3D classification was sufficient to select for ribosomes that contain the polymerase. So the big difference between the uncoupled and the NUS-G coupled expressome is, first of all, we can see NUS-G binding both to the ribosome and to the polymerase and to the ribosome. So it forms a physical bridge between the two machineries. But most importantly, it restricts the movement of RNA polymerase. So instead of having many individual clusters of RNA polymerase orientations, most of them now collapse into one cluster, which we originally suspected, this cluster 6. And that's because NUS-G restricts the movement that RNA polymerase can undergo. And so this is both because it holds it in a position where there would be steric clashes with elements of the ribosome if the polymerase were to rotate more, but also because the linker between the two domains restricts the flexibility that RNA polymerase can exhibit. So what might be functional roles of coupling? And so for this, we would like to make 2 suggestions. The first one is that NUS-G C-terminal domain, the very domain which binds the ribosome, recruits a number of different transcription factors, including termination factor Rho. But when it is bound to the ribosome, so bound to protein S10 shown in green here, then Rho termination factor is not able to bind NUS-G. And so we basically think, and we can confirm earlier proposals, that NUS-G when it is in the context of an expressome is not able to recruit Rho. So that means as long as you have a co-translating ribosome, transcription cannot be terminated. But as soon as the ribosome encounters either a premature or a canonical stop codon, NUS-G will be released, will be able to recruit Rho, and therefore transcription can be terminated. Another proposal that we'd like to make, which has not been made before, is that coupling of the polymerase to the ribosome through NUS-G also aligns the emerging transcript with ribosomal protein S3. And so this is important because S3 has been shown by Harry Noller's lab about 15 years ago to be part of the ribosomal helicase melting RNA secondary structures. And so what we think is happening is when NUSG couples the polymerase to the ribosome, the mRNA, as it emerges from the polymerase, gets directly fed into the ribosomal helicase and therefore is kept in a single-stranded conformation to avoid formation of hairpins, for example, which are known to inhibit both transcription and translation. And it's also interesting, if you look at an electrostatic surface on the ribosome, there is sort of a positively charged landing patch, if you wish, which is exactly where the mRNA is guided along before it's fed into the mRNA channel on the 30th subunit. So then the question what happens if the ribosome approaches RNA polymerase further to potentially form a collided complex? And so to do this, we used the same approach, but now shortened the distance between the polymerase and ribosome binding site. And we did this with and without NUSG. And so what we found now is another expressome state where the polymerase adopts a different orientation, one that very closely resembles the one that had been seen earlier by the Kramer laboratory. And here, the NUSG binding site on the polymerase and on the ribosome, as seen before, are too far apart. So NUSG can no longer form a physical bridge. And in fact, adding NUSG or leaving it out doesn't really make any difference, because the conformation that the polymerase adopts relative to the ribosome is now really driven by maximizing the surface complementarity between the two machineries and by minimizing the path length that the mRNA takes from the polymerase active site to the decoding center on the small subunit. And so Michael used a very neat approach to show this. He basically did an exhaustive search by generating all possible RNA polymerase orientations relative to the ribosome, and then looking which of those conformations are sterically possible. And when he highlighted the ones that are possible in blue, he could see a very close match with the particle orientations that we observe experimentally. Furthermore, if you plot the particle frequency shown in blue here versus the minimal mRNA length that is possible, you can see that the peak aligns with the minimal mRNA length or the minimal path length that the mRNA has to take in order to reach the decoding center coming from the RNA polymerase active site. In other words, we believe that the orientation that is observed in the collided state is largely driven by surface complementarity and by minimizing the mRNA path length. So with this, I would like to conclude and briefly walk you through the model that we would like to propose. So in an uncoupled expressome, the two machineries are really more or less independent of each other, and the only consistent connection is the shared mRNA. Once NUSG enters the picture, it restricts the movement of RNA polymerase relative to the ribosome, so that the nascent mRNA shown in purple here aligns with the ribosomal helicase, and we believe this ensures that it is kept in a single-stranded conformation. But when the ribosome approaches RNA polymerase further, a new conformation has to be adopted to minimize the path length for the mRNA and to maximize the surface complementarity, so you don't have any steric clashes. And so with this, I would like to conclude and really thank the people involved in this work. First and foremost, Michael, who is a postdoc in a lab and will probably soon start his own lab, so you should watch out for ads when he is trying to recruit students and postdocs. Maria is a research scientist in my lab who really started the project from scratch. We got a lot of help from Venki Ramakrishnan's lab, and in particular, I would like to thank the people at our EM platform, which, as you have heard from Bruno, is also an Instruct-ERIC hosting center. Without their support, we would not have been able to obtain these reconstructions. And we also got collected data at the ESRF and EMBL, and I would like to thank these funding agencies for their financial support. And I'm ready to take your questions. Thank you very much.
Thank you, Albert, for this great presentation. I was wondering whether somebody looked into the kinetics of RNA polymerase. Is the kinetics, the processivity of the enzyme modified by being within an expressosome and the different types of expressosomes that you showed?
Yes, so there is some evidence from, for example, Evgeny Nudler's lab, which suggests that a co-translating ribosome is able to mechanically push RNA polymerase forward. And this, might be relevant because the polymerase tends to regularly pause as it transcribes, on average every 100 to 200 base pairs. And some of these pauses are by RNA polymerase backtracking, where it basically reverses gear and moves backwards along the DNA template. and then a co-translating ribosome might then be able to push it forward and release the polymerase from this pause state. So this would be one example where you would have some kind of mechanical coupling between the two machineries.
There's some proofreading also going on, no? So would this prevent proofreading? And
so that's an open question and something that I'm very interested in addressing, because as you correctly point out, polymerase also backtracks when it makes a mistake. And so in that case, you would want the ribosome to prevent the mechanical push. You would like to prevent the mechanical push by the ribosome. So we don't know yet if the ribosome is able to distinguish between these different scenarios, but that's an open question that I would be very keen on addressing.
So there were several questions of the audience addressing about the same issue of does the speed of transcription or translation change the different stages of conformation stages? You answered that question, and whether all RNA polymerase conformations are functionally equivalent.
So I'm not sure about the last question. I mean, the polymerase can certainly adopt different. Oh, I see the different conformations relative to the ribosome.
Yeah, probably.
So we think that all these conformations in the uncoupled expressome, all the orientations that the polymerase can adopt, are functional. We don't have any reason to believe that one or the other would be inhibited by the ribosome. So yes, I think they're all functional complexes in that sense. Even if the polymerase and the ribosome are collided, we don't have as high resolution as we have for the uncoupled expressome for the polymerase part. But even in the collided state, we don't think that the polymerase itself is inhibited. So the ribosome obviously cannot further translate because it's blocked in its translocation ability, but the polymerase, we think, is still able to continue.
Mm-hmm. Sébastien Fribourg had a question about targeting this interface with drugs. Do you think it would be an interesting way to go to both target the polymerase and the ribosome?
Yeah, that's an interesting question. I mean, my feeling is that it would be very, very difficult. I mean, if nature has developed a lot of antibiotics that target either polymerase or the ribosome, but we're not aware of anything that targets the interface. I think it's a very dynamic interface. I think you would have to have a massive drug that has high enough affinity to block the interface. I'm not sure that's a very viable route. Mm-hmm. I also have to say that at this point, we don't know how essential coupling is. We know if we mutate the interface, in wild type E. coli, we are not able to get those mutants. But the problem is these interfaces are also involved in other processes. So we don't know if the problem is the lack of coupling or if it's because we mess up something else, like, for example, recruitment of transcription termination factors. So it's difficult to evaluate how essential the coupling is for viability.
Yeah, Nicolas Touras had a question related somehow, is wondering whether there is a model for the Bacillus subtilis species where there is no coupling and how this would affect viability or whatever. Yeah,
so all I know is that in Bacillus, the transcription rates are much faster than translation rates. And it's also, as far as I remember, there is very little row-mediated termination. So in other words, there is not this need to prevent termination factor row from accessing the naked transcript, if you wish. So one reason why Bacillus may not couple transcription and translation is because it may not have the necessity to prevent termination factor row from prematurely terminating transcription, if that makes sense.
Mm-hmm. So Adriana Ericamile out of the question where she was asking whether you add the single-stranded DNA to your system together with a minimum mRNA.
Of course, so we have DNA in our system because we form a functional transcription complex. It's not single-stranded DNA, it's in fact a double-stranded DNA that forms an artificial transcription bubble which anneals to the mRNA. And so, especially in our high-resolution reconstruction of the uncoupled expressome, where we see the active site of the polymerase in great detail, we know that it's a functional complex. The active site is able to bind a substrate, and it's a canonical post-translocated state, if you wish, waiting for the substrate to bind. So yes, DNA is there.
Okay, we have addressed most of the questions that were raised. Would like to thank all the speakers for this great session. I think it was very much oriented towards structure and function, but you understood also that there is a lot of interdisciplinary involved to characterize these complexities, to purify these complexities, to make these assemblies, and to solve the structure using the different approaches. This is really the essence of Instruct and what you want to promote with this organization. There was two questions at the beginning about training also in this center. So there are a number of workshops and hands-on training which are proposed by Instruct, and Pauline Aubergon, who is organizing this meeting, sent you a link where you can have a list of all the training opportunities within Instruct. Also, if you have a project which is accepted and granted, you may come and actually participate to data acquisition or data preparation, specimen preparation. So this is also an opportunity for you to be in such a center and see how things practically work. Okay. With this, I would like to thank you again for having listened to these talks and asked all these questions, and I hope it was interesting for you. Bye-bye.